does life come from? How do we begin? Thanks to advancements in technology and devoted researchers, we are learning more than ever about life on the microscopic level. Join me, Donna Diastole, as I ask the tough questions and delve into that most dark and mysterious of the human organ systems, once called the final frontier of the body, the female reproductive system. Tonight on Backline, early embryonic development. As you may be familiar with, human life begins with a single act of fertilization, when a sperm penetrates the outer layers of an ovum and their combination creates a unique set of DNA. But how does a single cell turn into amazing multi-billion-celled creatures such as you and I? To find out, we travel to Normal Heights, California, to the University of Duvall, where we find the female reproductive health specialist, Dr. Bruce Jensen and Dr. Steinia Ezimna, working in their state-of-the-art lab. Today is like any other ordinary workday for this dynamic pair of researchers, but beyond the fancy equipment and skilled technique, these two are pushing the boundaries of what we know about the first 3 weeks of our existence as an embryo. Good afternoon, Bruce, Steinia.
Welcome to our lab.
Good afternoon, Donna.
So, Steinia, after fertilization, when would you say the journey of the embryo begins?
Stientje Eisenche.
Excuse me?
My name, it is Stientje Eisenche. You said it wrong just now.
I'm sorry, Dr. Eisen. Ha! Ha! Excuse me. But where would you say the journey of the embryo begins?
Well, in humans, as in most other primates, fertilization takes place in the distal part of the fallopian tube.
And it takes about 4 days to travel along the fallopian tube and into the uterus. During this time, the single-cell embryo continues to divide into more and more cells, so that by the time it reaches the uterus, it is an advanced morula. But if you compare the sizes, the embryo is not much larger than it was when it started out as a single cell, because it is encased in a zona pellucida, which protects the cell from other spermatozoa after fertilization.
If you will have a look at the slides Dr. Jensen and I have prepared, you will see this for yourself.
Here we have a single-cell embryo not long after fertilization. Here you will see how it divides within the zona pellucida, and next a human morula from about 4 days after traveling through the fallopian tube, after it has just entered the uterine cavity.
Interesting. But what happens to the morula after it enters the uterine cavity?
Donna, before I answer your question, can I just say you're looking mighty fine in that turtleneck today?
Oh, well, thank you, Dr. Jensen. Now,
The way it compliments your eyes.
Dr. Jensen, what happens to the morula once it enters the uterine cavity?
What I think my partner is trying to say is that the morula develops an inner cavity and becomes a blastocyst once it reaches the uterus.
That's right. And the cells making a wall around the embryo will help to form the placenta and are called trophoblastos. These cells bring water, minerals, and amino acids from the nutrient-rich environment outside the embryo into the blastocyst cavity, where they reach the inner cell mass. The inner cell mass is called the embryoblast, and it will become the embryo itself. Now, Donna, if you want me to come over and look down my microscope, I can show you. Here we have an early blastocyst, and as you can see, here's the outer layer called the trophoblast. And there is a little cavity inside the blastocyst, And there is the inner cell mass. The zona pellucida still surrounds the whole thing, since this is an early blastocyst.
After the blastocyst forms, there are 3 more stages of development that take place during the first 3 weeks of pregnancy.
They are 1. The implantation phase, 2. The embryogenesis phase, and 3. The embryonic phase.
So tell me, what happens once we enter the implantation phase?
Well, Donna, after about 6 days since ovulation, implantation occurs, which means that little blastocyst gets rid of that old zona pellucida and sticks itself to the wall of the uterus, which has been preparing to receive the embryo.
The trophoblast cells that help attach the blastocyst to the uterine wall separate into 2 layers.
These 2 layers are the superficial syncytiotrophoblast and the deep cytotrophoblast. These will eventually form the embryonic membrane and the fetal portion of the placenta. The syncytiotrophoblast then grows into the uterus, and the uterine wall reacts by growing over the trophoblast, which concludes the implantation phase.
So the blastocyst attaches to the uterine wall, and then the trophoblast cells differentiate, and the wall grows over the outside of the trophoblast to bring the entire embryo into the uterine wall?
Yeah, this is correct.
So what exactly happens after the implantation phase?
Once the embryo is implanted inside the uterine wall, embryogenesis begins.
At the beginning of this phase, the embryoblast, that little inner mass of cells I talked about earlier, separates slightly from the trophoblast, creating a narrow space between them called the amniotic cavity. Here's what it might look like.
Uh-huh.
The embryoblast then flattens into an embryonic disc composed of 2 cell layers, the epiblast and the hypoblast. Some hypoblast cells multiply to form a membrane called the yolk sac. Now, this embryonic disc has 2 spaces, the amniotic cavity on one side and the yolk sac on the other.
Around day 15, the embryonic disc will develop a groove along the midline of the epiblast layer, called the primitive streak. This primitive streak will define the embryo's future right and left sides, cephalic and caudal ends, and dorsal and ventral surfaces.
The next step is gastrulation, in which the 3 germ layers are formed, all arising from the original epiblast. This process begins with multiplying epiblast cells, migrating medially towards the primitive streak and down into it. These cells replace the original hypoblast with a layer now called the endoderm. A day later, migrating epiblast cells form a 3rd layer between the first 2, called the mesoderm. Once this is formed, the epiblast is called ectoderm. I drew another illustration just for you, Donna. Epiblast cells, these white guys on the top here, migrate over the surface and down into the primitive groove, which is this guy right here in the middle. First replacing the hypoblast cells, these yellow guys down here, with the endoderm, then filling the space with the mesoderm, these pink guys right here in the middle. Once this process is completed, the layer formerly known as the epiblast, this top guy right here, is now called the ectoderm.
Thank you for that, Bruce. What happens after this?
Finally, there is the embryonic phase of development, which begins around day 16. During this phase, the placenta and other accessory structures develop, and the 3 germ layers begin to differentiate into their own respective organs and organ systems.
Now, tell me, what is the purpose of each of these 3 layers that have formed? The endoderm, mesoderm, and ectoderm.
The endoderm will later give rise to the respiratory system, the digestive system, and the bladder.
The mesoderm will later give rise to the kidneys, lymphatic system, cartilage, reproductive system, vascular system, muscle, bone, and the connective tissue.
The ectoderm will later give rise to the skin, hair, nervous system, brain, spine, and fingernails.
Some mesoderm overflows the embryonic disc and becomes an extensive extraembryonic mesoderm, which contributes to the formation of the placenta. And there is much more that we will not be covering today, Donna.
Thank you both very much for your thoroughly educating talk on early embryonic development. So let us put this in summary. Thanks to Dr. Jensen and Dr. Eisenach, today we were able to learn the stages of embryonic development. We discovered that there are 3 stages. First, the implantation phase, which occurs 6 days from ovulation. The blastocyst gets rid of the original outer shell known as the zona pellucida and trades it for a single outer layer of trophoblast cells. The trophoblast helps attach the blastocyst to the uterine wall, thanks to the 2 layers that it forms. The superficial syncytotrophoblast and the deep cytotrophoblast. Second, once the embryo is implanted inside the uterine wall, the embryogenesis phase begins. During this phase, the embryoblast, which is the inner cell mast of the blastocyst, separates slightly from the trophoblast, forming an amniotic cavity. The embryoblast then flattens into an embryonic disc composed of 2 cell layers, the epiblast and the hypoblast. The embryonic disc also has 2 spaces, the amniotic cavity on one side and the yolk sac on the other. The embryonic disc then develops the primitive streak, which will define the embryo's future right and left sides, cephalic and caudal ends, and dorsal and ventral surfaces. Next follows a very important step known as gastrulation, where the 3 germ layers are formed, known as the endoderm, ectoderm, and mesoderm, all of which give rise to very important human body systems during the final phase, known as the embryonic phase, which also includes development of the placenta and other accessory structures. It's safe to say that early embryonic development is fascinating, yet a very complicated process. Thank you all for watching and diving deep into the process of embryonic development with us. I'm Donna Diastole. Until next time on Backline.
The endoderm will later give rise to the respiratory system, the digestive system, and the bladder. Keep going. Just go. What do you think about that?
Just in the middle areas. Yeah, perfect, like that.
Sorry, it's like my pinky glue on your face.
My mustache.
Okay, alright.
Mustache tragedy.
That's right. And the cells making a wall around the embryo will help form the placenta and are called trophoblast cells. Stinja, I must ask you a question.
What is it, Bruce? Are you recording?
I've been recording all this time.
We'll shave it for later. When we're alone.